A modern glass skyscraper and a living plant face the exact same physical challenges. To stand upright and function, both require an outer protective skin, a structural filler to hold their shape, and an internal plumbing system to move water and nutrients. In plant anatomy, we categorize these three requirements into tissue systems. The outer skin is the epidermal tissue system. The structural filler is the ground tissue system, and the internal plumbing is the vascular tissue system. These three systems are constructed from basic cell types, like simple parenchyma for storage and complex xylem and phloem for transport. The way a plant organizes these cells determines the specific function and internal geometry of its roots, stems, and leaves. To understand a plant's survival strategy, we just need to look at how it manipulates the arrangement of these three structural systems across its different organs. Let's break down each system, starting from the outside. The epidermis is typically a single layer of compactly arranged cells forming the plant's outer boundary. On leaves and young stems, it contains pores called stomata, which regulate gas exchange and water loss. Each stoma is controlled by two specialized guard cells. The epidermis also produces specialized outgrowths depending on the organ. In roots, you will find unicellular root hairs designed for absorption. On stems, you will find multicellular trichomes, which help prevent water loss. Notably, while the stem epidermis is shielded by a waxy, waterproof cuticle, this layer is absent in roots, where surface contact is necessary for water uptake. Moving inward, we hit the ground tissue. This makes up the bulk of the plant body. Depending on the organ, ground tissue fills the spaces forming the cortex, pericycle, pith, and in leaves, the photosynthetic mesophyll. This diagram shows the different types of vascular bundles, which form the vascular system. When xylem and phloem alternate along different radii, the bundle is called radial. When they sit together along the same radius, it is conjoint. Everything inside the endodermis, including the pericycle, these vascular bundles, and the pith, is collectively called the stele. The geometric arrangement of these three layers, and specifically how the vascular bundles are grouped, is how we distinguish a root from a stem under a microscope. This image shows a transverse section of a monocot root. We can differentiate dicot and monocot roots by looking at the center. This monocot root is polyarch, showing more than 6 xylem bundles and a massive, well-developed pith. Dicot roots usually have a diarch to tetrarch arrangement, meaning just 2 to 4 xylem patches, and a very small pith. In both, the protoxylem points outward, an arrangement called exarch. Here we see a monocot maize stem sector. Notice how the vascular bundles are scattered throughout the ground tissue and surrounded by a sclerenchymatous bundle sheath. They are closed, meaning they lack cambium. A dicot stem, in contrast, features a neat, organized ring of open bundles that contain cambium. In stems, the protoxylem points inward, which is called an endarch arrangement. For leaves, dicots exhibit a dorsiventral structure with distinct palisade and spongy mesophyll layers. Monocot leaves are isobilateral. Their mesophyll is identical on both sides, and grasses often feature specialized empty bulliform cells that force the leaf to curl inward during water stress. Let's test your visual recognition. Look at this cross-section. Is it a root or a stem? Dicot or monocot? The scattered bundles with no distinct cortex or pith identify this as a monocot stem. The presence of cambium in the open bundles of dicots and its absence in monocots dictates exactly how these plants age. It is the biological mechanism that allows dicots to grow into massive, thick trees, while monocots generally remain thin. That increase in girth or diameter is called secondary growth. It is driven by lateral meristems and occurs heavily in dicot roots and stems, but is almost entirely absent in monocots. These diagrams illustrate the first step of the process. Strips of fascicular cambium inside the bundles connect with newly formed interfascicular cambium to create a continuous solid ring. This vascular cambium then rapidly divides, pushing secondary xylem, which we call wood, towards the inside and secondary phloem towards the outside. Look at diagram D, which shows a 2-year-old stem. As the inner wood expands, the outer epidermis is stretched and ruptured. To survive this, a second lateral meristem called the cork cambium, or phellogen, activates. It generates a protective periderm layer, replacing the epidermis with tough bark, while leaving small pores called lenticels open for gas exchange. Secondary growth is the plant actively replacing its primary tissues with heavily fortified secondary layers, allowing it to support a massive physical footprint. Synthesizing this gives a consistent anatomical logic. Roots have radial bundles and exarch xylem. Stems utilize conjoint bundles and endarch xylem. To differentiate plant types, a ring of bundles with cambium indicates a dicot stem. Scattered closed bundles indicate a monocot stem. Scan this QR code or click the link to download this complete cheat sheet. Keep it next to you as you practice identifying cross-sections. Mastering the relationship between these 3 tissue systems, from the protective epidermis down to the central stele, provides a fundamental framework for navigating the internal organization of flowering plants. Keep practicing these cross-sections, and I'll see you in the next session.